Well, I'm Daniel Ballesteros, but you can say Ballesteros. I'm used to. I'm working currently in Linnean Seed Bank with Dr. Hugh Pritchard, but I have been previously in different labs. And what I'm going to talk is about a work that I did in South Africa, trying to understand the mechanisms of cryoprotection of a particular type of plant, explant, that are the zygotic axis of recalcitrant seeds. Well, we all know that ice formation and growth is lethal for cryopreservation. Maybe not for ice cream making, but for us it's a problem when we go from the fluid state to the solid state in form of crystals. However, if we are able to manipulate our samples in a way that we produce an amorphous solid, I can call as a glass, we can have some success, and it's what some people, well, we have listened about vitrification and these kind of things. If we think in a seed, if we think in the physical chemical states in a seed, we can see through this graph that, for example, at room temperature, as you reduce the water content, you can enter into a viscoelastic, a solid amorphous state. You can enter into a glass when you lower the water content below 1.15 grams per gram. In the case of desiccation tolerant materials, so the orthodox seeds, this happens naturally when they dry on the bed stump or in the ambient conditions. Or it can happen in a seed bank where we also go lower when we dry them to water contents around 0.05, and then we lower the temperature to -20 or to liquid nitrogen temperatures to favorite a longer term preservation. But with this kind of desiccation tolerant orthodox material, we don't have problems of ice crystal formation, vitrification, devitrification, cryocrisis, and that kind of things, because the materials is always in a solid state. It's always in the glass form. However, we have different types of plant materials, as the previous speaker has commented. There are plants that don't produce orthodox seeds. There are plants, for example, that produce recalcitrant seeds. Recalcitrant seeds are high water contents, and they are sensitive to desiccation. So if you lower the water content below 0.3, and just remember this graph, if you remove the water content below that water content, they die. So you cannot preserve you cannot induce a glass just by desiccation as you do in the orthodox seeds. But I mean, we can cryopreserve recalcitrant seeds. Different researchers during the last 25 years, they have been able to cryopreserve seeds at water content between 0.3, 0.5, and ice formation is still possible, but with fast cooling and different things, you can allow some success. The problem is that the success is species dependent and is explant dependent. You can also preserve shoot tips, as my colleague has presented. You can take from the plant the shoot tips and do the cryopreservation processes. With recalcitrant seeds, if we select the zygotic embryo, it's just because you can have more biodiversity, because each seed produces an embryo, so you can have more diversity, and also because with just one zygotic axis you have the shoot and the root meristem, and you can have a whole plant without using a lot of hormones and a lot of in vitro culture. So with desiccation tolerant seeds, we only have to dry to reach a glass within the cell, and we can lower the temperature. But with the desiccation sensitive material, with recalcitrant seeds, we are in an area, in a gray area, where we have to do something to avoid ice crystal formation. So what people has been doing to avoid that is trying to desiccate as much as possible without losing viability, trying to to use appropriate cooling rates, in this case, really, really fast cooling, and trying to protect against oxidation during the whole process from the excision to the recovery. That is a big problem, as the previous talker has mentioned. But how this happen? What are the mechanisms that are allowing success and cryoprotection? So in the early 90s, it was shown that by cooling really, really fast, we could increase the survival. What bottom is the? What bottom is the red point? Maybe this one in the middle. Okay, yeah, great. So by drying really, really fast, it was detected that we could cryopreserve materials at water contents as low as 0.3 grams per grams, while if the desiccation was slow, you couldn't preserve things at water contents below 0.8. When measuring the water properties of these tissues, if these water properties were changed during these two type of treatments, they realized that there were no changes in the water properties. So what was the mechanism that was allowing cryopreservation? Research that was performed after that has concluded that most of the protection that you get by drying really, really fast, and because you can dry to lower water contents, is because by drying really, really fast, you avoid an intermediate water content where most of the oxidation occurs. This has been tried to be improved by the use of antioxidants. For example, the use of cathodic water that was developed a few years ago by the lab of Patricia Berjac and Norman Palmenter, and trying also to minimize the excision damage during the excision process. So in the case of shoot tips, I know when working with battery pens in Cincinnati that they had problems depending on at which part of the stem they were excising the axis, and the more mature and the more immature, and the time that it needed from the first axis excised to the last one. So there are a lot of parameters that can be in the case of zygotic axis, also depends on if your excision is really close to the shoot tip, or if you allow a little bit of cotyledon around, like a protective area. So there's a lot of work trying to improve this oxidative damage, because we know that is one of the mechanisms of cryoprotection, trying to minimize this type of oxidation. The other thing the other mechanism that was detected is that by doing a fast cooling, this type of sharp peaks that you can see here in samples that were cooled very slow, those sharp peaks were completely absent in the samples that were cooled really, really fast. Those sharp peaks have been related to water, to free water that probably is in the wall, in the cell wall area, and not inside the cells, because but the fast cooling didn't affect or didn't change any of the thermal water properties. So the amount of water at which ice was detected was still the same, and the way that this water was frozen, the enthalpy of melting was also not changed. Another interesting thing was detected is that the fast cooling by the use of cryo-electron microscopy, the fast cooling was able to maintain the structure, the histological structure, the cytological structure when cooling fast compared when cooling slow. When cooling slow, all these type of valleys and things that can be seen around were related to ice damage, to the ice growing and damaging the cells. But when cooling really, really, really fast, there was not this type of ice damage, and it was also related in different research to the formation of a glass. So summarizing a little bit, the mechanisms for minimization of desiccation and freezing stress that has been detected till now is that we have to minimize the ice crystal growth by formation of a glassy structure, usually by fast drying and rapid cooling. We have to minimize the oxidative stress by fast drying and chemical protection, and also by the management of the explants. But any of the water freezing and melting properties have been affected by doing this. So, but as I said at the beginning, that all this cryopreservation in plants, in recalcitrant seeds, is species dependent, and some species they do it well just by drying. Some species they need the use of cryoprotectants in order to be success. But how this happen? What are doing cryoprotectants that drying is not doing? And how can we use all this information to improve our methods of cryopreservation? So there are only a few words trying to understand these mechanisms. One was this work in 2006, Christina Walters and their work. They detected that the use of PBS2, the plant vitrification solution that my previous speaker has introduced, by the use of this solution, you are able to reduce the amount of water inside the cells, so to reduce the amount of water that can form ice, and also you are able to create a glass transition. You can detect a glass transition, so you are able to make a glass below 120 degrees. So the use of this plant vitrification solution also affects to the freezing and the melting properties of the explants, and it affects them by increasing the amount of water that you can have in a tissue before you detect ice, and also it changes the melting, the enthalpy of the melting by reducing this enthalpy of melting, and as that means that anything that goes farther to 330 millijoules per milligrams of dry weight, that is the enthalpy of melting of pure water. Anything that goes below that, it means that it's highly concentrated, that even if you can find water that crystallizes, the system is highly viscous, highly concentrated, so probably the vitrification is going to be better. So similarly, this previous study was done in shoot tips also, and this was also done in shoot tips, and by using cryo-SEM and differential scanning calorimetry, this team from Spain, they detected that in the different steps of the plant vitrification solution cryopreservation, you are reducing the water, so the damage by water, they said that these black pockets here and were down to water, to ice crystal growth, and you can see that the cytoplasm is completely destroyed during. And as you lower the explant with more and more cryopreservants, and then you cool it fast, you create a structure that is like a glass, and also is related to a glass transition temperature about minus 44. So if we summarize these two words and also other words that I modified the presentation in the train, but I couldn't upload the new presentation. So there was another slide with a work that Hugh Pritchard has been doing with PBS2, but on recalcitrant seeds, and they also see that PBS2 reduce water content and produce a glass at about minus 40, minus 50. So summarizing the mechanisms of cryoprotection by this plant vitrification solution is that reduce chemically, it makes a chemical desiccation and reduce the water content, and for so it reduces the probability of ice crystals growth. It forms a glassy structure, and the water freezing and melting properties are largely affected. So by PBS2, you can freeze safely samples at higher water content. But all this work with PBS2, where we more or less know the mechanisms, has been done mostly in shoot tips that they are very at high water contents. When we think about recalcitrant seeds, PBS2 it has not been used very successfully. This desiccation and the use of other cryopreservant has been more successful. And we are also at lower water contents than these plants. So the question in my research was how the cryoprotectants and the desiccation that people is using recalcitrant seeds, how are they affecting and modifying, what are the mechanisms of cryoprotection? In order to study these questions, I used a differential scanning calorimeter to see calorimetry, to see if there were changes in the water behavior and the intracellular ice formation during the melting and freezing of these samples with different cryoprotectants, glycerol, sucrose, a combination of both PBS2 and different ones, and also using cryo-electron, scanning electron microscopy, but with a cryostat. I wanted to see what was the frozen state of the samples after the whole preservation process. So the samples were treated as normally, they were plugged in liquid nitrogen, and then directly moved into the scanning electron microscopy, and they were always below 100 degrees. So we got a quite good idea of what the structure looked like. I selected 3 different plants, 3 different species. One was a species that has a very small embryo, and it has a really good response to cryopreservation without the use of any cryoprotectant. The other one was a species that has a good cryo response, but you need glycerol to have success in the cryopreservation, and also have a really small embryo, and the desiccation rate of these 2 samples is quite similar, so they were comparable. And also I used a explant that was a higher embryo. You needed to use part of the cotyledons to avoid excision damage, and the response was not really good, and also glycerol was needed. So first, when comparing the water properties of these 2 explants, the one that has good cryopreservation just by drying and cooling, and the one that needs glycerol, apparently there were no differences in the water properties in fresh material and in material that had been treated with glycerol, sucrose, PBS2, or different. Most of the amount of water that where you could detect ice in the DAC was quite similar compared with the rest of recalcitrant seeds, and only there were significant differences, but very, very small, when you were using glycerol as a cryoprotectant. There was a small increase in the amount of water that you could have inside the cell before ice could be detected. This increase is not as large as the one detected with PBS2, where you had this increase in water content to about 0.6 or 0.7. So it's not that large, the effect of glycerol compared with the effect of the whole PBS2. And there were no significant changes in the enthalpy of melting. So it means that any of the cryopreservants is changing the internal. The other thing we saw is that in the species that needed glycerol for cryoprotection, you could detect this type of sharp peaks that has been associated to free water, probably free water around the cell walls, and you cannot detect this in the species that just by drying you can cryopreserve it. The other interesting thing that we observed with the DAC is that while in the species that needs glycerol for cryoprotection, at the water contents where cryoprotection was successful, we just observed this type of peak that it was reduced with glycerol. So as we also can see here, the scan was quite plain, and even if there was a small of water freezing, it was not damaging for the structure. However, in strictness, in the one that just by drying and cooling you have success, you can see that before this amount of water that of course is reduced with glycerol, you see this kind of vitrification, devitrification event, and it's in some way it's saying that the structure is entering in a glassy state in a natural way without the use of anything, even at that high water content. So with all these things on mind, we went into the scanning electron microscope. We wanted to see if these small things we were watching with the DSC were also reflected in the structure. So summarizing all the photographs and everything that we made, you have in this column images of the sample when they were not dry. So they were at the higher water content, 2 grams of water content per gram of the weight, and they were plunged into liquid nitrogen and moved into the scanning electron microscope. And these samples here, they were dry to the water contents that are the water contents where we have success in cryopreservation, and we just freeze them really fast and put them in the cryostat. What for us is interesting is that the drying process, it produces, of course, a shrinkage of the cell, but you have these large vacuoles that they don't change the volume, and they are keeping all the cell in some way there. Okay, and the other thing is that you can see that the connections between cells, so the cell walls connections, and they keep also very well. So all the tissue rings together and the structure is keep very, very, very well. I didn't put the image, but with other samples, the castanospermum, this one that is a big embryo that we didn't have success, you could see that all the structure collapsed a lot because they didn't have this type of large vacuoles keeping the cell apart. So something was needed to avoid that collapse. All right. And well, you can see that this large vacuole is occupying the center of the cell. So we go to the species that needs glycerol for cryoprotection, that the desiccation is not enough, that you have that kind of sharp peaks of free water. And when you dry the sample, you can see that the cells are completely distorted and they shrink a lot, and the volume is changed a lot. And not only that, you can see these large holes that are formed between cells, and all the tissue is coming apart. And so probably the external walls, the area between walls in the tissue is full of water, that when you remove it and you continue drying, it creates this kind of distortion. And how is glycerol avoiding that? Because we have seen with the DSC that is increasing slightly the amount of water that the tissue can have to freeze it, but that's all we have seen. What we saw using the scanning electron microscope is that when you infiltrate, when you allow the tissue to take this glycerol, the morphology or what we see is completely different if we compare these images here with these images here. We can see this kind of net that is occupying all the cells and all the spaces around, and interestingly, this kind of net follow this pattern. Sometimes here that you have inside these vacuoles, there is this kind of reticulus, that kind of pattern. We think that these kind of small balls you can see all around are ice clamps. Wesley Smith suggests us that they were ice clamps. He has a lot of experience in this, and you can see that when treated with glycerol, you have all this net around the cell that probably is the glycerol that is going into the whole structure. And when drying to the water content that is optimal for cryopreservation, we have a different picture, because what we have is that even if the cells are shrinking, they are shrinking all together, and all the structure is shrinking all together, and we don't have those huge spaces. We measured the same with sucrose. We saw different response, and in glycerol, in Carnospermum australe, what we did was another technique. We got a sample, we treat it with glycerol, we dry it, and then we rehydrate it, and we cut it with a microscope. And what we saw is that samples that were not treated with glycerol, just by the drying and rehydrating process, the cells were being damaged. You can see that the membrane goes apart from the wall. As you can see, depending on you were using confocal or a stain, you can see that the membrane is shrinking. The cell is shrinking while the cell wall is still there. Another thing is interesting is that the morphology of the cell gets that kind of roundy shape. The samples treated with glycerol, we don't see this kind of collapse of the membrane. We see that the membrane is all, the apoplast is all pasted into the cell walls, and the back walls are around. They are not kind of in the middle. And the other thing is that you can see that the morphology of the seed is still hexagonal, and so in some way what glycerol is doing is protecting the structure during cooling, drying, and during recovery. So with all these results on mind, we can say now that we know a little bit of what are the mechanisms of cryoprotection in zygotic embryos of recalcitrant seeds, differently than the mechanisms of cryoprotection when using PBS2. We have water content reduction, but this time is mainly by drying, not by chemical desiccation. We have reduction of ice crystal formations, mainly by drying and fast cooling. There's a facilitation of a glass formation, as we have seen in the strychnos samples, but also as has been measured in other samples in previous research. And differently than PBS2, we don't see a large effect in the water freezing and melting properties. But what we see is a strong stabilization of the cell structure during cooling and drying. I know that, in order to afford a maximum cryoprotection of zygotic embryos, we have to take into account all these conclusions. We have to inhibit ice crystal growth, minimize oxidative stress during excision and processing of the samples, facilitate a glass formation, and try to stabilize the cell structure during cooling, during drying, and also during rewarming. Probably these mechanisms are different than the mechanisms when cryopreserving shoot tips with PBS2, and probably are different than the mechanisms when cryopreserving animal cells. And also they are species dependent sometimes, but by knowing these mechanisms in the different species, we may be able to anticipate how to preserve the variety of plants. And we are talking about more than 10,000 species with recalcitrant seeds around the world, most of them with interest in agriculture. And I just would like to thank everybody in South Africa who helped me during my stay there, and also to Professor Hugh Pritchard that has permitted me to come here to explain a little bit this research. Thank you very much.